Let us consider the process of DNA replication using a circular bacterial chromosome as an example. The replication of linear chromosomes is going to be very similar, with one prominent exception that we will discuss later. And so here we have a double-stranded DNA molecule, and the outer circle is one strand and the inner circle is another strand. DNA replication starts at positions in the genome known as origin of replication, and the origin of replication is the site of the assembly of a huge complex of proteins called the replisome. We are not going to discuss at this point details about the replisome, but instead discuss the process of replication. And so we have, once the replisome has assembled, what will in fact happen is that this double helix is going to get unwound. So it's going to look something like this. And so you can imagine this unwinding process as sort of unzipping the double helix, and this unzipping of the double helix is going to simultaneously progress in both directions. And these locations where the unzipping is occurring are known as replication forks. Let us look at what happens at a replication fork in greater detail. So let's say we are focusing on the left replication fork. And so perhaps the top strand is 3' on the left and 5' on the right, while the bottom strand is 5' on the left and 3' on the right. And this is the fork, the replication fork, and this replication fork is moving to the left. In order to initiate the process of DNA replication, we need an RNA primer, and these RNA primers are synthesized by this enzyme called primase. And the RNA primer is going to be anti-parallel, since it base pairs with the template strands is going to be anti-parallel, and so 5' in this primer is going to be on the right and 3' is going to be on the left. And similarly, in the RNA primer on the template strand that's on top, 5' will be on the left and 3' will be on the right. Next, once the primer is in place and makes available the hydroxyl group at the 3' end for the RNA polymerase to come and carry out the polymerization reaction with the triphosphate to make the phosphodiester bond, the RNA polymerase is going to come in and it's going to start to synthesize the daughter strand in the 5' to 3' direction on the daughter strand, and Therefore, when you're copying the template strand on the bottom, you are going to be synthesizing from right to left, whereas in the top strand you are going to be synthesizing from left to right, because that's the direction of 5' to 3' synthesis on the daughter strand. Now, after a while, the replication fork would have moved further to the left, and so our DNA molecule is going to look something like this. And we have 3' here, 5' as before, 5' and 3', and this is the location of my primer where synthesis had been initiated previously. And my polymerases have run ahead, and this is the daughter strand on having been synthesized from the bottom template strand, and the polymerase that's copying the daughter strand from the top template strand is over there, and this is the daughter strand that has resulted from copying the template strand on top. Now, for the strand that's being synthesized on the bottom, the direction of movement of the polymerase is the same as the direction of movement of the fork. The fork is moving to the left, so is the polymerase. And as the polymerase approaches the fork keeps the replication fork keeps moving to the left. That means the double-stranded DNA keeps getting unzipped, exposing more template DNA, and the polymerase will basically run a circle around the entire circular DNA molecule. And this kind of synthesis is relatively straightforward, and we call this leading strand synthesis. The daughter strand that's being copied from the template strand on the top, however, does not work like that, since the polymerase is moving to the right, which is not the same, or which is the opposite in the direction in which the replication fork is moving. And what that means is that new DNA in this purple segment, new DNA or template strand, has been revealed or exposed that was not available previously, that was in a double-stranded form previously, and no daughter strand has been synthesized. So if this situation is not corrected, it's going to be problematic, since the daughter cell will not get its full complement of double-stranded DNA, and therefore what happens in this situation is that a new RNA primer is synthesized, and a new daughter strand is made in the 5' to 3' direction with a new polymerase molecule. And this implies that the synthesis of the daughter strand on the top is fundamentally different from the synthesis of the daughter strand on the bottom template strand, and it has a special name. It's called lagging strand synthesis. And one result of lagging strand synthesis is that the daughter strand is being synthesized in fragments, and these fragments are called Okazaki. Fragments. Now, it is worth pointing out that lagging or leading strand synthesis has nothing whatsoever to do with 3' or 5', or whether you're on the top or whether you're on the bottom. One and only one thing determines whether you have leading strand synthesis or lagging strand synthesis, and that is whether the direction in which the DNA polymerase is moving is the same as the direction of the replication fork. If the polymerase is moving in the same direction as the replication fork, then you have leading strand synthesis. On the other hand, if you have the DNA polymerase moving opposite to the direction of the replication fork, then you end up doing lagging strand synthesis. And the situation looks something like this, where over here we have the template strand, and here is the DNA, the new DNA that has replaced the RNA primer, and over here we have the end of the Okazaki fragment. And there is no phosphodiester bond between them. And when you have DNA where there is no phosphodiester bond on one of the strands, that's called a nick, and that situation is unstable and needs to be avoided. And we also know that DNA polymerase 3, which normally carries out the synthesis, requires there to be 3 phosphates, a triphosphate, in order to make the phosphodiester bond. And we don't have a triphosphate here. We only have a single phosphate group. And therefore DNA polymerase 3 cannot make the phosphodiester bond. Instead, there is a special enzyme called ligase that is able to make a phosphodiester bond between two nucleotides, neighboring nucleotides, where one of them is not a triphosphate but a monophosphate. And therefore the ligase comes in and makes the phosphodiester bonds and connects up the Okazaki fragments with the DNA that has replaced the RNA primer, and thereby synthesizing a continuous stretch of a daughter strand in lagging strand synthesis when the DNA polymerase moves in a direction that's opposite to that of the movement of the replication fork. Next, let us consider how you can get errors during replication and how those errors are corrected. The nitrogenous bases have tautomers or isomers. In which the hydrogen atoms or protons have been moved around, and the nitrogenous bases can readily interconvert between the normal forms and the tautomers. However, the alternative tautomers are rare and are not common. So, as an example, we can consider cytosine, and if you move this hydrogen atom to the nitrogen in the third position from the nitrogen that is bonded to the fourth carbon, then you get the imino form of cytosine. And the consequence of this imino form of cytosine is that whereas normal cytosine base pairs with guanine, the movement of the hydrogen atom to the nitrogen in the third position allows this imino form of cytosine, this C asterisk, to base pair with adenine instead of guanine. And what that implies is that sometimes, rarely, you will make mistakes when you are replicating DNA. For example, let's say our template strand is ACC TGA CGG from the 3' to the 5' direction. So when we synthesize the daughter strand in the 5 to 3 direction, we will get TGG AC, and normally one would get T here. However, since C asterisk, the imino form of cytosine, can base pair with adenine, occasionally we will get a situation like this. You will get the imino form of cytosine base pairing with the adenine, and therefore introducing an error in the DNA. How are these errors corrected? The polymerase has two abilities that allow it to fix these sorts of errors. The first ability is that it can proofread. So DNA polymerase is a proofreading enzyme, and it can actually detect when a mismatch has been made. The second ability that the polymerase has is it's got what is known as 3' to 5' exonuclease activity. A nuclease is an enzyme that can digest or cut nucleotides, and an exonuclease is a nuclease that works from the outside in. And 3' to 5' means that normally a polymerase moves on the daughter strand in this direction, 5' to 3'. This is the normal movement of the enzyme. However, if the enzyme is correcting a mistake that it has determined through its proofreading ability, then it has the ability to move in the 3' to 5' direction and act as an exonuclease to cut or remove this mismatched base pair. And so it moves back to this position, and then it can begin the synthesis afresh. To summarize, because of these tautomers of the nitrogenous bases, rarely you can incorporate the a mismatched base pair, because those tautomers, they can make hydrogen bonds or base pair with the wrong partners. However, the DNA polymerase does proofreading, so it can detect these mismatches, and then it can move back. So it can move in a 3' to 5' direction to excise through its exonuclease activity the mismatched base pair and put in the correct base pair. And because of this, DNA polymerase is highly accurate, and it makes one mistake in 1 billion base pairs. Because of the proofreading and 3' to 5' exonuclease activity, DNA polymerase is a highly accurate enzyme which makes only one mistake in copying 1 billion base pairs.